Okay, so I'm going to go through section 5.3 and I think 5.4. We didn't finish chapter 5 notes today. So we left off talking about the components of phospholipids and how it's made of a glycerol neck that interlinks phosphate groups into fatty acid tails. We talked about how membranes form spontaneously because the fatty acid tails are nonpolar, and so nonpolar substances tend to hang out with each other. And then describe the factors involved in membrane fluidity, which just means that some organisms have the enzymes that can add double bonds to their fatty acid tails and put that kink in them. That makes them more fluid when temperature changes, or they can make themselves more rigid. So 5.3 is going to look at functions of membrane proteins and illustrate all the different jobs that proteins can do, and some of it will be review. Talk a little bit about how proteins associate with the membrane, and then define transmembrane domains. So proteins can perform some key functions, probably 6 important jobs for the cell. They can be transporters, which we're going to talk more in depth here today and when you come back from the weekend, but they're very selective. Substances enter and leave through these channels or carriers that are made of proteins. A second job is with enzymes, which means they attach to the membrane, and their main job really is to carry out reactions. These two kind of go hand in hand. Cell surface receptors assist in chemical messaging. They receive molecules from the outside world and transmit them to the inside of the cell, maybe to turn on some genes. Cell surface identification markers, non-self versus self. Are you part of the organism or are you a foreign substance? Cell-to-cell adhesion means that they can glue themselves to one another. This could be permanent, so like a group of similar cells working together, or this can be temporary, maybe during the process of development. And then finally, attachments to the cytoskeleton and how proteins help anchor the cytoskeleton to the cell, and maybe to help with interaction of other cells. So this is a slide that shows the 6 different functions. So here's my selective transport. Certain molecules travel through it. This is to represent enzymes that carry out chemical reactions. Cell surface receptor. So this molecule will dock and maybe transmit a signal. Here is our cell surface identification marker or identity marker with glycolipids and proteins, cell-to-cell adhesion, and then finally that attachment to the cytoskeleton. Now, way back in chapter 3 when we were talking about macromolecules and the 4 levels of protein structure, we talked a little bit about how when we were discovering proteins, it was really hard to figure out exactly what they did, but we did notice that they had some similarities among one another. We call them motifs, reoccurring thematic structures that appear, like beta sheets and alpha helices. I gave the prime example of of motifs in arts, like how Michael Bay always uses explosions, or in James Bond, he always says the name's Bond, James Bond, shaken, not stirred, or whatever. So same thing with proteins. Proteins are very diverse in what they do and the shape that they come in, but they do have some common structural features. And so one common structural feature is that they have to anchor themselves to the lipid bilayer. So we talked briefly yesterday the difference between peripheral proteins and integral proteins, and how peripheral proteins will latch on to the outer surface or inner surface of the plasma membrane. These peripheral proteins do not transverse or go through the plasma membrane at all. They're on one side. They're either on the outside or the inside. And so one common feature that they have is that there are nonpolar regions that are inserted into the plasma membrane. And so I love this diagram because here's this peripheral protein and it's anchored by the use of sticky fingers or greasy feet. And I just love the term greasy feet because these fatty acid tails are nonpolar. And nonpolar means that you're a wax or you're an oil. And so grease belongs in that category, and they just describe them as feet. So they're like literally embedded into the plasma membrane. We also have proteins that go all the way through the plasma membrane, so we call them integral membrane proteins. They go through, and so the common structural feature that they have is that the interior portion of that protein has to be nonpolar. And so we're going to see some nonpolar amino acid alpha helices as well as beta-plated sheets. But if the interior is nonpolar, so it can interact with these fatty acids, well, the outer portions of these integral proteins have to be polar because the phospholipid heads are phosphate groups, and they are polar. So we have polar interacting with polar and nonpolar interacting with nonpolar here. Okay, so another common structural feature that we find is the number of times it can cross through a membrane. So really that's what we call transmembrane domains. They differ in how they cross the membrane. So here we have a single pass protein. Here we have multiple pass proteins, but the main difference is how many times does it cross the membrane to anchor itself to that membrane. And then the final common structural feature we find are pores. Pores made up of beta barrel proteins. As you can see here, a bunch of beta barrels linked together to form this very large complex channel so molecules can travel through. So a review question, membrane proteins are distinguished by what chemical property? That is A, they possess regions of hydrophobic amino acids. Okay. Next review question, the specific function of a membrane within a cell is determined by the, that's D, type and number of membrane proteins. The transmembrane domain of an integral membrane protein is made up of hydrophobic amino acid, does or can form alpha helical structures, can cross the membrane multiple times. So really, it's D, all of the above. So that was 5.3. Moving on to 5.4, looking at simple diffusion and facilitated diffusion, differentiate between channel proteins and carrier proteins, and then talk a little bit about osmosis. So passive transport means that we are going to move molecules from high concentrations to low concentrations without using any energy. And how we're not using energy is because we're moving down the concentration gradient. We're moving from high to low. It's kind of like when you're at the top of the hill and you want to go sledding. You know, you just give yourself a little push and you let gravity do the job. You're not really using any energy. It's because you're moving from high elevation to low elevation. So passive transport doesn't really use any energy. We're moving from high concentrations to low concentrations. And there's 3 ways that cells use passive transport. Simple diffusion, which just means that they're literally going to transverse or cross the plasma membrane. Facilitated diffusion, which means we're going to be using protein channels and protein carriers. And then the third way is osmosis. So simple diffusion means we are moving substances from regions of high concentration to low concentration. So when you see brackets, brackets mean concentration. So high concentration to low concentration. So one thing we have to keep in mind with diffusion is that molecules, ions, atoms, they're always in random constant motion and they always want to reach equilibrium. So this net movement of molecules will continue until the concentration is the same in all regions. So you can kind of see in this picture down here that I have this molecule with a lipid bilayer and it has the ability to cross through it. And we're moving from high concentration to low concentration until we reach equilibrium. And when we reach equilibrium, movement is still occurring. Like this molecule can move back across the plasma membrane and this molecule can move in this direction, but there's still movement occurring, even though we have this major barrier. Now, this major barrier is limits what molecules can cross, and so we call it limited permeability or selective permeability. So I'll talk a little bit about which molecules can cross the plasma membrane. But protein channels, okay, channels and carriers allow the membrane diffusion to be selectively permeable because these channels and carriers can be specific. So they're specific as to what they latch onto and move across the membrane, and this is really important because it can move some really important molecules like glucose or sucrose or ions. So channel proteins have a hydrophilic interior, which means they love water, provides aqueous channel that polar molecules can pass when they open. Carrier proteins bind specifically to the molecule they assist. So there's this hydrophilic center in this, right here, and carrier proteins latch on to specific molecules. Now size, charge, and rate of diffusion across the membrane can differ between different molecules. So if a molecule is hydrophobic, it can pretty much pass through easily because the fatty acid tails are hydrophobic, and you see this arrow is big and kind of fat and bolded. Small uncharged polar molecules like water, glycerol can pass through, but you can see that the arrow is not as big as these guys up here. Large uncharged polar molecules, you see that they just bounce right off. Same thing with ions because they're supercharged, so they really rebound off the plasma membrane. Okay, so with channel proteins and carrier proteins, we can have ion channels, which just means that they are specific to ions. So ions are atoms that have a charge. Some channels are gated, which means that they can open and close in response to a stimulus, maybe a chemical stimulus that they receive or electrical stimulus. Well, we see a lot of gated ion channels in our nervous system. This is actually how a nerve impulse is sent. So there are three conditions that determine the direction of this movement: relative concentration of the ions on either side of the membrane, a voltage difference for gated ions, and then is the gate open or closed. So you can kind of see that this neuron, if we zoom in on it, you can see it's negative on the inside and positive on the outside. And when a nerve impulse is sent down, the charges literally flip, kind of like a wave in a stadium. People stand up and then they sit back down. Well, the same thing with the charges. The charges will flip-flop. So this positive becomes a negative, and this negative becomes a positive, and it sends a wave down the axon, carrying a message, from some type of stimulus. So that's all I really want to chat about for ion channels. Okay, facilitated diffusion means that we are using proteins to help us move substances from high to low. Could be a carrier protein or a channel protein, but carriers bind specifically to a molecule. And once all of those carrier proteins are occupied with the molecule that they are moving, the rate of diffusion will not increase. In fact, it will plateau out because there's no more carrier proteins to, latch onto that molecule and move it. As you can see with this graph, simple diffusion, which means they can just travel through the plasma membrane, if you keep increasing the rate of if you keep increasing the concentration of whatever you're moving, well, then the rate of diffusion is going to increase as well. And there's really no limit. There's really no limit as long as you have one side that's high and another side that's low. Whereas with facilitated diffusion, once all those carrier proteins are occupied, moving the molecule that it wants to move, then it plateaus off. Okay, so I've talked a little bit about simple diffusion. I've talked about facilitated diffusion, specifically with ion channels. And then the third way for passive transport is osmosis, where we're going to move water from high concentrations to low concentrations. So one thing I need you to understand is that when you see a picture like this, and you see all these little dots, these represent solutes. So over here, I have a high solute concentration. When you have a high solute concentration, then you need to assume that you have a low water concentration. So I have low water on the outside, high water inside the cell. So water always moves from high to low. So water will leave the cell. And as a result, instead of being a nice donut-shaped red blood cell, the red blood cell shrinks and shrivels up because water is moving from high to low. It's leaving the cell. So we call that hypertonic solution. And one way I remember hypertonic solution, where the cell shrivels or shrinks, is that small children are hyper. And so think of the cell as a small child. It's small, and hypertonic solution. So yeah, that's one way I remember hypertonic solution means the cell is going to shrink. Excuse me. Now, hypotonic means that you have a solution that has low solute concentration. I did not mean to go on to the next slide. So I have low solute concentration. As a result, I have high water concentration. So I have high water out here, low water inside the cell. Water moves from high to low, and as a result, the cell will increase or swell, and it gets really, really big. So we call this a hypotonic solution, and hypo sounds like hippo. Isotonic means that the concentrations inside and outside the cell are pretty much relatively the same. And so we have movement. We still have movement of water from leaving the cell and entering the cell. They are at equal rates, and there's really no change in the cell size, but there's still movement. Okay. Aquaporins are water channels that specialize in movement of water. They were realizing that the rate of the cell size when it changes due to water wasn't happening as fast as it should. And so they're like, something must be assisting this, and they discovered aquaporins. So aquaporins were just recently discovered, I'd say within the last 15 years. But aquaporins, their main job, move water. They are protein channels that move water. Now, cells of prokaryotes, fungi, plant, and protists have strong cell walls. And so when water enters or leaves the cell, we have the cell wall that gives it additional support. So here, in an animal cell, when water rushes in a hypotonic solution, if the cell takes in too much water, it can burst and lyse and basically destroy itself. But here with a plant cell, because we have this cell wall, it gets targeted and provides support, and the cell will not rupture. Here with a hypertonic solution, the animal cell will shrink or shrivel, But here with this plant cell, it just gets plasmalized. So it just wilts. And if you just add some water to it, then it perks right back up. Isotonic solutions, we just describe them as flaccid. There are 3 ways that organisms can help maintain osmotic balance. And the first one's called extrusion, which means that we have these vacuoles that specifically take in water and then release them. And then they take in more water and then release them. So you can kind of think of it like you're in a boat and you have like a small little leak of water, but you have a bucket. So then you're just like scooping the water out and dumping it over the side. So that's called extrusion. And this happens in single-celled eukaryotes. We also have osmotic regulation. You see this with oceanic creatures where they adjust the solutes inside themselves to match the outside environments. So that way there is no net flow. There's still water moving across in equal rates, but we're not losing or gaining anything. And in plants, they have turgor, which just means that they have this central vacuole that results in that turgor pressure. And they do depend on that turgor pressure for support or shape. Review question: which variables influence whether a nonpolar molecule can move across a membrane by passive diffusion? That would be B, the difference in concentration of molecules across the membrane. Which of the following does not contribute to the selective permeability of a biological membrane? We talked about this, we talked about that, and we also talked about the hydrophobic barrier. So D, it's hydrogen bonds. We have not mentioned hydrogen bonds at all here. The movement of water across the membrane is dependent on the solute concentration, not the solvent. Water doesn't have a carrier protein, so it's the solute concentration. If a cell is in an isotonic environment, then which of the following is true? So isotonic means that they will stay the same size. So you're like, okay, it's either going to be B or D, but this means that no water will move across the membrane. Well, that's false. There's still movement, but there's no net gain or loss. So D is your more correct answer. And then I put this question at the wrong section. Okay, so then we took notes on active transport. So active transport means that we are going to move molecules from low concentrations to high concentrations. And in order to do that, we have to use energy. Usually that energy is in the form of ATP. It does involve some highly selective protein carriers that have 3 classes. So there are 3 classes of these. We have uniport, which means it will move one type of molecule across the membrane. We have symports, which means that we will move 2 molecules in the same direction. So you can see S1 and S2 are moving inside the cell, or maybe they're going to leave the cell, but they're moving in the same direction. And then we have an antiport protein carrier that carries 2 molecules, but in opposite directions. So one will enter the cell and one will leave the cell. And this is pretty much where we left off. So I guess on Monday we will finish the notes for Chapter 5. I will post the lab, and then we'll carry out that lab Tuesday and Wednesday, and that will be it for college biology before MEA break.